Okay, well, thank you everybody for taking time out of your schedules to join us and find out about the new and exciting features in IST 6.5. I am Pantelis Vlachos, and I'm a biostatistician in the Cytel offices in Geneva. And I will be presenting the first half of today's presentation, going through the methods that are behind the enhancements that we have added into IST. I will go initially in transition from IST 6.4 to 6.5, telling you about what has been added, what's different between the two versions, the three new modules that we have added, while my colleague Charles will also refer to some further enhancements in addition to providing you with a demo of the software. So going into IST 6.4, this is the current version of our software, for which I'm sure that quite a few of you are already familiar with. And this is the structure of the modules currently in IST 6.4. Within each one of these modules, there are enhancements that Charles will go over towards the end of today's presentation. But the focus of today's presentation is going to be three new modules that we have added. One having to do with population enrichment, one with program-level design, and MCPMod. So let's begin by going first into the MCPMod module, which deals in general with dose response studies. The goals of these studies is to establish proof of concept and see basically how a change in the dose results in a desirable change in the endpoint of interest. And the second goal is a dose-finding step, is one that tries to select one or maybe more than one good dose levels so that it can be passed over for a confirmatory phase 3 trial once proof of concept has been established. The current approach, the traditional approach, does this in two separate steps. So the proof of concept is conducted using multiple active arms and placebo. You have the selection of the target dose, which is the statistically significant dose at the proof of concept stage, and actually picking up the smallest of such doses, but also the one that is clinically relevant. In the dose response modeling step, data that have resulted from the proof of concept step and from earlier trials are being used, and a statistical model that captures the effects of the target dose of the dose response is being utilized. So this is a straightforward approach. It has some drawbacks, however, and mostly the drawbacks have to do with the fact that this method tends to concentrate the range of doses studied to a narrow range where the sponsors can have faith that it will establish a clear dose signal. It is also it's clear that the dose response model should itself play a greater role in choosing the right dose. But, however, with the current approach, the dose response modeling comes in at the very end of the process, and a much better approach would be to try and design the trial with possible statistical models in mind. So for that, the MCPMod methodology has been developed and has been around for quite some time, and it consists of two separate components. If you view them individually, these components, the MCP component basically treats dose as a qualitative factor and then proceeds in making inference about the target dose restricted to this discrete set of doses that you used the trial. On the other hand, the mod component uses a parametric functional relationship to model the relationship between dose and response. In other words, it treats dose as a quantitative factor, and the validity of the modeling approach depends on pre-specifying appropriate dose-response models. So these two components with the MCP methodology are actually being put together, okay? And they construct the MCPMod methodology. The difference, I'll be explaining essentially the difference in the traditional pairwise comparisons by this use of the dose-response modeling. So at the design stage, we have a pre-specification of candidate dose response models. Each of these dose response shapes in that candidate set is going to be tested using appropriate contrasts and employing MCP techniques, multiple comparison techniques that will preserve the type 1 error. Now, proof of concept is going to be established when at least one of the model tests is going to be significant. And once this proof of concept is established, either a best model, single best model, or a weighted average of a set of significant models is going to be used to estimate the dose-response profile and the target dose of interest. So, in other words, MCPMod combines multiple comparison and model-based approaches. It tends to be robust to model misspecification, and it is quite flexible in dose estimation. So it results overall in more informative designs, and which form a solid basis for a confirmatory study, and they have been endorsed by the two major regulator agencies. MCPMod is not new to Cytel and to our products. For the analysis part of the software, Cytel has already introduced a couple of years ago Proc MCPMod, which is already available in the SaaS platform. What you will find in E6.5 is this new module that, besides analysis, it also includes the design, where you also have sample size and power, which are produced individually for each candidate model, but also in some summarized fashion. This design can be based on optimal allocation, and it is currently available for normal, binomial, and count endpoints. So this is the first of the three enhancements that we have made to the software. The second one has to do with population enrichment, in particular adaptive population enrichment. I'm going to be using a motivating example for this, just to provide the setup for the method that's currently being implemented within yeast. So angiosarcoma is an ultra-orphan disease. It has been poorly addressed by current treatments. Pazopanib is a VEGF inhibitor that shows modest benefit, but TRC105 can actually complement pazopanib by inhibiting endoglin, which is a different antiangiogenic target. An adaptive trial is considered to be optimal for this type of case because this refers to a small population. We have about 1,800 cases a year. We have limited prior data. And for this particular case, there seems to be subgroup interaction because there appears to be a greater benefit possible for the addition of TRC105 to pazopanib for cutaneous versus visceral tumors. So the type of design that has been employed in such a situation is the following that you see here, and which I'm going to try to explain in the graph that you see in front of you. So we have an interim analysis. So first of all, we have the enrollment in two cohorts that will be done in a sequential manner. So accrual in the second cohort will only start after the first cohort accrual is going to be over. So the interim analysis that we have here is going to be based on patients from the first cohort only. Okay, the first cohort, these are patients, all comers, who have been obviously randomized in either pazopanib alone and pazopanib plus the addition of TRC105. Now, if the treatment shows significant improvement than the standard of care into the full population, so if we are up here, both in the subgroup and in the complement of the subgroup, no changes should be done to the trial. The second cohort will recruit the pre-specified number of subjects from both the subgroup and the complement. And then the trial will end when the required number of events from the two cohorts are going to be observed. One can think of this scenario as the full population as being in a favorable zone. So this entire design can be thought as an extension of the current sample size re-estimation procedure that is already available in yeast, which is done based on the promising zone methodology. Now, if the treatment does not show significant improvement but shows promising improvement when compared to the standard of care, increase the sample size for the second cohort. Again, the second cohort will include patients from the full population, and users should actually modify just the study duration in case of treatment is promising in the full population. So in that situation, we are in this part. Now, let's go to the further down. If the treatment is not doing better than the standard of care in the full population, okay, but it's only doing better in the positive subgroup, then the new treatment is performing well. This means that there is a clear interaction between the biomarker positive group and the new treatment, and therefore, for the second cohort, we will only recruit patients with the positive biomarker only. Okay? So this scenario is when the full population is unfavorable, but the subpopulation, the biomarker positive subject, is favorable or promising. The last scenario will be when the treatment does not work in the full population as well as the positive population. This is the case when the new treatment is unfavorable in both full population as well as the subgroup. And we also have an option to stop for futility in case results are are beyond a threshold for a futility stopping. So this is the adaptive study. As you can see, there are two, as we mentioned, there are two cohorts. It's a two-stage design, so there's going to be a p-value from the data from cohort 1. There's a p-value resulting from the data from cohort 2. And we're going to be ensuring that type 1 error is going to be preserved by performing close testing. So the close testing uses, obviously, a combination function and a condition on the combination function, whether we are actually in the case of no enrichment or whether we are in the case of enrichment. So these particular designs are available in E6.5 for time-to-event endpoints. With time-to-event endpoints, we also have some special challenges because in order for this methodology to work, you need to have independence between the p-values in the two stages, between P1 and P2. We cannot use auxiliary data in the sense of observations of cohort 1 that become completers in cohort 2, because using auxiliary data essentially destroys the independence of P1 and P2, and this has been demonstrated in the literature. So instead of choosing the interim analysis time point to split the data in cohort 1 and cohort 2, what needs to be done is to pre-specify the allocation of the events and the sample size to each cohort before taking the interim analysis. So, for example, as an example here, you can specify that cohort 1 is going to be 70 patients and 60 events. The interim analysis is going to be taken after 4 events have arrived, and cohort 2 is going to contain 54 subjects corresponding to 35 events. This allows for a full inspection of all cohort 1 data at the interim analysis, including the censored observations. And this is evaluated through simulation. This is a simulation-based approach, which means that you will get to tune your design by selecting different options, different combinations of patients and events for the cohorts, as well as the point at which the interim analysis is going to be taken. So to conclude and to recap the type of a design, we have initially the enrollment of subjects in the full population. We have the interim look. We can either proceed according to the favorable zone in the an unblinded sample size re-estimation setup where we are in the favorable zone and there's no change in the second cohort. If we have promising results, we proceed with a sample size increase in the full cohort. And if we are in the unfavorable zone, we can either fall in unfavorable for both the subpopulation and the full sample size, which means that we cannot perform any change in the second cohort, or if we are favorable with respect to the subgroup, then we can increase the participation of the subjects from the subgroup only. So this is the second module that has been added, and my colleague will actually, Charles, in the second half, will also present you with more a demonstration demonstration and more options on how this is actually being implemented in EAST and what the output looks like. In the third and final module that we have added, we are dealing with program design. The motivation of this was actually given to us in a project that we had to deal with. This was a situation where a sponsor was preparing an early development plan, and he had posed the questions on how the relationship between early biomarkers and clinical points can be leveraged to optimize phase 1-2 plans. In other words, to improve the quality of information or the speed of development time, and how data from the phase 1b data can be used to optimize the dose selection for a phase 2b study, allowing to reduce sample size and improve chance of picking the correct dose to pass into phase 3. So the kind of plan that was envisioned can be seen in the schematic here. So this the plan, the whole development plan started with a biomarker proof of concept trial that you see here on the left, which involved just a high dose versus the placebo. Then there were go-no-go criteria based on Bayesian analysis of the data to compute posterior probabilities of a response for the two biomarkers that were considered in this case. And if we had a go scenario, then we go to a biomarker dose-finding trial, add some doses, and get the new biomarker information. And then we have another set of go-no-go criteria that will allow us, if the criteria is go, to move to phase 2b with the clinical endpoints and leverage that information from a combined analysis of the first two trials to help design a phase 2 trial with the clinical endpoints, or to stop. And then for the phase 2b trial, there's another set of go-no-go criteria that allows you to move into phase 3 and a phase 3 dose choice. The key objective of that design that we had in that case was to design the phase 2b trial to try and maximize the probability of getting the phase 3 dose right. Now, here the way that we approach this through a program design simulation, it includes simulating, first of all, clinical trials themselves. So in each one of those steps, you're actually simulating clinical trials. We are all familiar with the concept, and this is sort of like an overview of what clinical trial simulation involves, which is to define the true underlying scenario for each one of the endpoints, study designs, and the decision rules that you have, generate as many repetitions as you can, summarize the results, compare them across the scenario and the possible choices, and use those simulations to choose and justify the trial design. What is newer is drug program simulation. In drug program simulation, we not only we use all these individual clinical trial simulations, or the sequence of those, and the decision rules and design options for moving from one trial to the next. Our aim here is to optimize the sequence of trials for a particular set of drug program objectives. So in EAST 6.5, we have implemented that for two types of drug program sequences. One is in the dose escalation, where the first stage is a standard dose escalation design, one of the already available dose escalations designs in EAST. Once the maximum tolerated dose is being obtained, then we switch to a single-arm cohort expansion, to an efficacy endpoint, and we use frequentist or Bayesian go-no-go rules. The second type of program development has to do is taken from the area of oncology, where we have a phase 2 oncology trial followed by a group sequential design. So the first stage can either be a single-arm binomial, Simon's two-stage design, or a two-arm survival. The second stage is a group sequential design, and the overall probability of success of the program is being reported in the end. Schematically, and this is the example for the first of the two cases that I mentioned, you would go, once you have selected, for example, a 3+3 design on the left for your dose escalation study, and once the MPD has been obtained, you would go to a frequentist base on the right, go-no-go study, single cohort expansion with an efficacy endpoint in mind. So these are the three areas in which we have added modules and have expanded the capability of EAST. And my colleague Charles will actually go now and illustrate how these modules work within our software.
Okay, thank you, Pantelis. Hi, everyone. So I'm going to try to translate some of the concepts that Pantelis has presented into how we're going to implement this into EAST software. And EAST 6.5 right now is still in beta testing mode. So I'm not going to do a full live demo, but I'm going to show you some screenshots so you can visualize how the key inputs and outputs are going to look like for these new modules. So in the first demo, I'm going to jump back to MCPMod. And in this example, there are going to be 4 active doses. So if we include the placebo, there's going to be essentially 5 doses, 0, 0.05, 0.2, 0.6, and 1. And we're going to do a one-sided, well, it's going to be by default one-sided, but with alpha is 0.05, and we want to compute the power for a given sample size. So here I've chosen a sample size of 100, and if we allocate these 100 patients equally across the 5 arms, that's 20 patients per arm. And we'll choose a common standard deviation of 1. And the power of the design is going to be a function of the individual powers from each candidate model. So remember, each candidate model will generate an optimal contrast with power to detect proof of concept, and the power of the overall design can be summarized by one of these functions, mean, minimum, maximum. So I'm going to just choose mean for this example. So that's the test parameters. And I'm now going to jump to the allocations, and there are a few different options here. So you can define your own, right? The user-defined allocations, and by default they'll be balanced. Or you can try to optimize the allocations, and we'll do this calculation for you. There are a couple of different criteria, such as the D-optimal or the target dose, which is defined by the delta here, or a combination of both. So that's what you're seeing in the screenshot, and when you click optimize, EAST will tell you, what is the optimal proportions or ratios to satisfy that criterion. But for simplicity, in this example, I'm going to stick with our original user-defined allocations, which is just balanced. Okay, so of course in MCPMod we have to specify the candidate models, and here we offer the standard choice of the many different model families, and for each we have to specify a placebo and max effect, which usually would be the same across all of your candidate models, as you can see here. And for each candidate model, you need to specify the model parameters, of course, and these are the standardized versions of the models. And you can see I've added a bunch of candidate models here. If I click on this icon, I can blow up the plot. We can see the full plot of the all 6 candidate models here. There's a linear 2 different versions of Emax, a beta, and 2 logistic functions. Okay, so with everything specified, it's just a simple matter of clicking compute, and it will compute this is the mean power of the design. And if we look at the individual power for each of the 6 models, that 37% is essentially the mean of these 6 individual powers. Plenty of other outputs from this, but just to give an example, we can look at the optimal contrast that's computed for each model. shown either as a matrix like this or as a plot. And some other details. Well, first of all, let's try a different example where clearly 37% power is not going to be sufficient. So we want to be able to calculate the sample size required for, let's say, 90% power. And so simply we select this button here, sample size. We enter our desired power of 90%. Okay, and we're going to keep everything else the same in the candidate models and allocations. And again, clicking compute will generate this sample size of 469 patients, which is around 94 patients per arm if we divide them equally across the doses. So both ways we can calculate the power from a given sample size or sample size for a given power. Okay, so that's very quickly the MCPMod demo. Let's move on to the population enrichment example. And this is going to be similar to the case study that Pantelis presented. I'm going to try to simulate it as close as I can in EAST. So in this trial, we have a binary biomarker where around, you can see the subpopulation prevalence here, around half of the population is assumed to be biomarker positive. And well, actually, before we jump into population enrichment, we would first, use EAST using standard methods to determine the sample size and events required. Let's say for 90% power. So assuming that before jumping into population enrichment simulation, we've determined that we need, this is a total of about 135 patients and 110 events to obtain our desired power. So that's going to be our constraints to begin with. And so the sum of these two cohorts will come from a previous design. Now, this standard group sequential method may work well if the population were actually homogeneous. But suppose that the treatment effect in this case is going to be not as strong in the biomarker negative patients, right? So we have, you can see the hazard ratio here of 0.65 compared to the biomarker positive patients 0.55. So the idea, if you remember from Pantelis's flow chart, is that we're going to do an adaptive enrichment design where at the interim, if we find the results from the full population are not favorable or promising, then we can enrich and focus solely on this subpopulation of biomarker positive patients. So that's the idea here, and there are a lot of moving parts to this design, and so I'm just going to give an overview of some of the key decisions that you need to make when trying this. And we have some nice graphical tools to help you. So this is one example. The first set of decisions is how to split the sample size and events into two independent cohorts. Okay, so if you look at, if I go back to one previous slide, we're splitting the sample size of 135 into two cohorts. So as soon as you come to the 81st patient arriving, that they're defined as cohort 2. And similarly, we need to split the total events into, how many patients, how many events we're going to observe, before switching. So the general idea is to maximize the efficiency of the design. We want to split the cohorts in such a way that the target events in each cohort basically are reached around the same time. So the green curve here is the events from the first cohort. We're targeting 70, and the black curve is the events, the total events in both cohorts, and we're targeting, I think it was 110. And you can see that reached around the same time. So that's good. And we also want to minimize the number of patients at risk in each cohort. So that's in the first cohort, that's the gap between the events and the sample size here, right? The green and the red, and the gap between this black curve, the events, and the total sample size, this purple line here. So that seems to be this pattern looks pretty good. Okay, and if not, we would go back to our tab. In fact, it's actually the same tab. I'm showing you in two different slides, but on the same tab, you can fiddle around with the split of the sample size and events while preserving the total. Okay, so now the other decision is how to time the interim analysis, and there's similar heuristics that you might use in group sequential or adaptive designs. Basically, you want the interim to be not too early in the trial. Okay, so if it's too early, there's not enough events to make an accurate assessment of the treatment effects. If it's too late, then you're going to reduce your potential savings. And in the case of an enrichment trial, what this means is, by the time, if you have an interim that's too late, you're going to have enrolled already, and you decide to enrich, I should say, then you may have already enrolled, many biomarker negative patients who are not going to be followed up. So in this case, we're going to place our interim close to the end of, cohort 1, where cohort 1, cohort 2 split, which seems like a pretty good compromise between, the two extremes being too early or too late. Okay, in the next tab, adaptation parameters, this is where you define your promising zone. So there is still a standard promising zone design built in here, where you can, so even focusing on the full population, you can decide to increase sample size and events. And then in the bottom half, these are the parameters relating to adaptive enrichment. So you'll enrich if the full population is not favorable or promising, and you can define all these boundaries here. So, if you decide to enrich how many patients and events, so you can, let me read this out here. You'll basically enrich if the conditional power for the full population is less than 30%, right, which is defined up here. So that's unfavorable for the full population. And the conditional power for the subgroup is better than 50%. So that's enough to enrich, and you can also specify a futility boundary if you're going to fail on both full population and the subgroup. So all of those parameters are specified in this tab here. And in the interest of time, I'm going to jump straight to the output. So if you run, say 10,000, 100,000 simulations with all different scenarios, this is just one particular example, may not be the most interesting example, but I'm just showing you what the kinds of outputs that you want to look at. So we're breaking down all the simulations by zone, right? So at the interim, you're going to fall into one of these zones. And let's just look at the conditional power on the far right. So when when the results of the interim are favorable, of course, conditional power is going to be high, so that's fine. If you enter the promising zone at the interim, you're going to increase the sample size. Maybe this increase is not enough, but it looks like your conditional power is 70%, which is like 68%. Which is okay. Now, in a standard promising zone design where we don't enrich, and you end up in the unfavorable zone, you're typically going to have low conditional power. But because we've built in the adaptive enrichment rule, if we decide to enrich, we can have 66% power compared to the cases where you do not enrich is 47%. So these are the sorts of key outputs that you're going to be assessing and you want to do this obviously for a range of different scenarios, not just the one that I've shown here. Okay, so in our final demo, I'm going to show you an example of the program design module. And as Pantelis mentioned, there are two types of programs. The one I'm going to show you is the first, where you do a dose escalation followed by cohort expansion. The other type you can do is doing a phase 2 oncology followed by a group sequential design. So in this example, we're going to begin with one of the phase 1 dose escalation designs as part of the escalate module. And here I've chosen a MTPI, or in fact, we can now do an MTPI-2. And this is the decision table from MTPI-2, and of course hidden in all these tabs, if you're familiar with this module, are the standard design parameters, so the sample size, any prior information. You can build in different probabilistic stopping rules, and you need to specify a dose toxicity curve. So once you've done all of that, you can add a cohort expansion phase and simulate an efficacy endpoint, which could be normal, binomial, or survival. And in this example, I'll pick the binomial single proportion, right, such as a response rate. And notice that this says a go-no-go rule, right? But basically, a go-no-go decision is going to be made at the end of each simulated cohort expansion. So, and, the simulations, each will be tracking each of these decisions. So, in the cohort expansion phase, we're going to add 20 patients. Who will be given, basically at each simulation the MTD that was selected in the phase 1, and then we're going to simulate efficacy endpoint and make a go-no-go decision based on these rules here. So, as Pantelis mentioned, we can do either frequentist or Bayesian. So if this were Bayesian, we can specify a prior, and these go-no-go rules are based on posterior probabilities. So in the screenshot you see here, we're going to go if we're 80% confident, better than 80% certain that the rate is better than 0.35, and we'll stop. No-go decision is made if we're less than 10% certain that the rate is better than 0.45. So you can see the cutoffs are shown here as a function of the sample size. So the green is the go cutoff, the red is the stop cutoff for a given observed response rate. In between the cutoffs, okay, so if you fall somewhere in between, then you, of course, you have you're in the consider zone, which means you need more information before making a definitive conclusion. So now that we've specified both the phase 1 and the cohort expansion, that's our one scenario defined, and we can simulate this or different versions of it. I'm just doing one particular scenario, but you may want to do different scenarios with different go-no-go rules or different sample sizes, for example. And there are a lot of different outputs. The top table here is just showing you, some like what was the true MTD based on our assumed dose toxicity curve. So it was 25, which is dose 4, and the MTPI-2 does a pretty good job of selecting dose 4 as the MTD most of the time. Well, the most frequent, so about 36% of the time, and otherwise it almost always selects one of the neighboring doses. So that's fine, and let's look at what happens when we add the cohort expansion and with a go-no-go decision at the end. So given the particular efficacy curve, we almost always end up in a go decision, and we can see how this is broken down by different doses. So remember, our true MTD was dose 4, 25, and when that is selected, we make a go decision almost 100% of the time. If we happen to select a lower dose at a given simulation, then you can see we're making go decisions less frequently and more considered decisions and no go decisions. So that's all very intuitive. And okay, so that was a quick demo of the 3 new modules. In addition to the new modules, we're going to be including many enhancements that customers have requested in the last couple of years for our existing modules. So modules that are already available in 6.4, we're basically sort of upgrading them with some new features. So I'll just quickly go through each of these. The base module, we're adding a super superiority design, which is commonly used in vaccine trials. And the multi-arm, multi-stage module, we're going to be expanding different endpoints, so you can do using a binomial endpoint, you can do the multi-stage group sequential, and a survival endpoint, you can do a p-value combination. So filling in some of those gaps there to make it much more flexible. In the sequential module, we're adding equivalence designs, so you can basically do an equivalence trial with group sequential boundaries. For ADAPT and SERVADAPT, which implements our promising zone designs, you can now do sample size re-estimation for non-inferiority. Predict is our module for event and enrollment prediction, and in 6.4, Predict relied on piecewise exponential simulations, and now we've added a Weibull distribution for simulating events. As I showed you in the program design demo, Escalate, we're adding a few new features, including the MTPI2, which is becoming more popular as a replacement for MTPI. And in endpoints, the endpoints module, you're going to be able to design trials that have mixed endpoints, so combining, for example, normal and binomial endpoints. And there's going to be a neat graphical tool to create different types of testing procedures. So thank you very much, and we'll be happy to take your questions.
Hello, and this is Mike White together here in Cambridge with Charles and with Rishikesh. And before we begin the question and answer portion, I just want to thank everyone for joining today. We're very pleased with the response, and we'll begin the question and answer period. I'll be posting select questions. You can submit your questions simply using the Q&A panel, and we'll get to as many questions as the time allows. I did want to remind everyone, of course, that you can, for a limited time, download the slides from today's session right from our events page, and that's simply cytel.com/events. You'll also see in that page places where you can experience East 6.5 and all the modules in East coming up. The first one would be, of course, we'll be back at JSM this year in Vancouver, and that begins in just a little less than 2 weeks. This year, our own annual statistics symposium will be hosted by Merck KGA in Darmstadt, Germany. That will be November 14th and 15th, and the November 15th was actually a dedicated East Training Day. So to find out more information about either JSM or our statistical symposium this year, just go to the events page. Again, www.cytel.com/events. Maybe to begin the questions, we'll pose one that some people have experienced before and have been posed before, because Cytel, and there may be users of our Prox MCPMod that have actually joined us, and I'll ask about the Prox MCPMod and the differences. And either for Charles or for Pantelis, how would you describe the difference between Prox MCPMod, which of course is associated with SAS, but how is it different from the new module of East?
Well, I can take that, Mike. So the PROC only does the analysis part of an MCPMod. The procedure that you see now, the module in East, will actually do the design part as well. So you will do the powering and the sample size estimation needed for a particular power for each one of the models that is being entertained, as well as in the summary of these models.
And another question that occurs, and maybe this is for Pantelis as well. Pantelis, you mentioned during your presentation that the FDA and EMA effectively, I think you used the term endorse the new methods, population enrichment, and multiple comparison procedures. Maybe you can elaborate a bit more about the experiences or the perspective of regulators when utilizing these methods. I do believe that these methods have already been implemented in ongoing trials, but of course, maybe you can tell us more about that.
Right. So my comment referred to the MCPMod in terms of being endorsed by the EMA and the FDA. And as you have mentioned, we have had experience within Cytel, actually, with implementing in ongoing clinical trials MCPMod methodology. The couple of cases that I have been involved with deal with the analysis part. But I think, well, I'm not sure if we had the design part yet within Cytel, but I have attended in different conferences and most adaptive type of meetings, presentations in which, especially given by colleagues at Novartis, in which this type of methodology has been applied in their trials of their own, the design part as well.
Thank you, Pantelis. Of course, we hope so many of you will be interested to know when to expect E6.5 and the new modules. When will they be actually available for users? Charles, maybe you have some insight into that.
Sure. Thanks, Mike. So we're planning right now an early fall, so in September sometime should be ready to be shipped.
And I might add that, of course, users, people and companies with current license of these will, of course, be alerted when the actual release is ready for customers, and we'll alert everyone who is on our WebEx today with us, and in fact, all those who have registered. We're now looking at the questions from users, from the folks who have joined us today. So Okay. So we see a question here from Vanessa Rupnik. Charles and Pantelis, does the software now have the capability to predict multiple events per patient for endpoint modeling? So Pantelis, maybe perhaps you can address this question from Vanessa.
Sure what is meant by multiple events. I'm assuming that it refers to the predict module, which is currently used for prediction of events. We haven't, like the addition that has been made in the predict module, or the change is that we are now allowing for the Weibull distribution to be the one that is used to generate observations rather than, well, in addition to the piecewise exponential, but I'm not quite sure what is meant by multiple events, by predicting multiple events.
A question now we have from, I believe the name is Hayoung Zhu. Maybe for you, Charles, is the sample size calculation function implemented in MCPMod?
Okay, thanks for the question. I'm not quite sure I understand it. Maybe you could elaborate over the chat, but I think the answer is yes. In that, in the demo that I showed, we can calculate both ways. Given a particular sample size, we can calculate the power, and for a given power desired, we can calculate the sample size.
Thanks, Charles. Hayoung Su also asks, as the MCPMod is now available in EAST, will the SAS proc be further developed and supported? And we're polling our experts here to say a little bit more about that. Rishi, would that be something you might want to answer?
Yes. So we do have a plan to continue developing proc MCPMod, especially for our SAS users who want to carry out, the entire analysis using SAS, and they can stick within SAS and continue using MCPMod. So yes, we will be continuing and supporting proc MCPMod, even if there is a module in EAST.
And maybe further, can the MCPMod be used for sample size calculation?
Yes, MCPMod, the new module in EAST, can definitely be used for power and sample size computation. As Charles showed, it can be done using optimal contrast and allocation method.
Thank you, Rishikesh.
Mm-hmm.
And we'll still have a minute or two left, so if anyone would like to pose more questions, we'll keep our eye out here. We can continue for a few more minutes until we get to the top of the hour. Here's a question: can the MCPMod be used for different endpoints?
Yes. The plan currently is to release MCPMod for normal, binomial, and count endpoints in E6.5. So yes, it can be used for different endpoints.
And again, while we're selecting questions and monitoring your questions, I'll remind everybody that that these slides today will be are available right now on our events page. Also, the replay for today will be available by Monday. And again, of course, everyone who's registered, everybody who's joined us today, will alert you to that availability. And of course, we certainly endorse and hope that you'll share today's presentation with colleagues. Let's see. We're looking at additional questions. Stand by as we see here. Yeah, Chitra Taradakyo Vlachos, sorry for the... maybe Rishi, you could respond directly. Or
yes, so Chitra, I think the difference between MTP, I think Pantelis will be able to shed more light on that. I'm not.
So, what is the question? Can you repeat?
The question from Chitra Pantelis is how is MTPI2 different than the MTPI design, the original MTPI design?
All right. Okay. So the MTPI2 design is also based on former Bayesian decision theoretic framework, and it's adjusting for Occam's razor. So it mitigates some of the suboptimal decisions that MTPI makes and provides a theoretically optimal decision rules. So it provides more refined actions that allow more efficient exploration of different doses in the dose-finding process. Within the actual publication from the MCPI2, you can actually find more details about it, but basically it hinges on user-provided probability quantities. One is sort of like the target probability of toxicity, and epsilon 1 and epsilon 2, which define the underdosing and overdosing regions. So it treats any dose with toxicity probability smaller than the probability of toxicity minus the target in the underdosing region, or larger than the probability of toxicity plus epsilon in the overdose region, as being lower or higher than the MTP, respectively. So there is an optimization that is done through a decision theoretic framework that the authors have made.
Thank you, Pantelis. And with that, we're at the top of the hour, so we're going to conclude today's session. Once again, I'd like to thank everyone who's joined us, and of course, our presenters today, Pantelis Vlachos, Charles Liu, and Rishikesh Kokone, who is our customer champion, shall we say. So thank you once again. Please watch for upcoming webinars to talk more about some of the modules you've seen today in EASE, and we'll be dedicating webinars in the near future to the specific modules. And of course, we want you to join us there, and we really appreciate those users out there, and the feedback that we get from users is really an important component of the development of EASE. So please keep your, we take that feedback very seriously. It's an important aspect of the progression of EASE. So once again, thank you for everyone joining us today, our presenters, and I wish good day, good evening to all. Thank you.